All right. So today we are going to talk about the autonomic nervous system. From the pharmacology point of view, okay, from the pharmacology point of view, general pharm point of view, because that's something I've also had to explain quite frequently in the last week. Let's get started. The autonomic nervous system. When you think of the autonomic nervous system, you should think of the automatic control of the body, the part of the nervous system that controls or regulates body activities automatically, almost with an automated kind of response. And there are two components of that system that we are interested in: the sympathetic system and the parasympathetic system. The sympathetic and the parasympathetic nervous system. If you don't know this particular area, it's going to be very difficult for you to master drugs and to figure out what medications you need to treat X disease or Y disease. Now, when you see a presentation in a person related to autonomic nervous system, many times people are going to present with either heightened sympathetic activity, in which case, if you're going to manage this patient, you either want to inhibit their sympathetic activity or increase their parasympathetic activity. Okay, so think of the body like a balance, a beam balance, right? We have this balance going on between the sympathetic system and the parasympathetic system. And I'll explain what each of them is in a bit and how you should probably picture them to remember them in the long term. If someone has a lot of, let's say, increased heart rate, increased contractility, constipation, they can't pee, those who constitute sympathetic kind of presentations. So you're looking at the vignette, you're looking at this patient, he has all these sympathetic responses or sympathetic presentations. How do you treat this patient? They're going to ask you for the management, or they're going to ask you for the mechanism of action of the medication you're going to use to treat this patient. Your first thought should not be to try to just memorize one medication from somewhere. Your first thought should be, okay, what do I need to do in this patient? I recognize first, this is a sympathetic overdrive presentation. So what we are going to do is either try to decrease or suppress the sympathetic activity or to try to increase the parasympathetic activity. It's like when you have two people on a scale, if this one is too heavy, one thing you can do is to decrease the weight here to maintain the balance, or you can increase the weight over here and you also maintain the balance. And that is how we figure out medications and therapies when it comes to the parasympathetic and sympathetic pharmacology. You want to bear that in mind. So what does sympathetic activities look like? And what do parasympathetic activities also look like? Sympathetic activities, simply put, is everything that happens, that you experience when you see a lion. Think of it. As a person, when you see a lion, how will you respond? That's what some people call fight or flight, right? Imagine everything that you can think about. Just think about it. It's very simple. Let's think about sympathetic actions. Sympathetic activities. Your heart rate is going to go over the roof. Your eyes are going to open up. Your pupil will dilate because you need to see where you are about to run to, right? So you see pupillary dilation, which we are going to call mydriasis. Your sphincters are going to be short. Why? This is not the time to pee. This is not the time to use the bathroom, right? So all sphincters are going to be short as well. You may have increased sweating. Sweating is a little complex, maybe both sympathetic and parasympathetic at the same time. We have all these activities going on as part of the sympathetic response. Your heart beats faster. It also beats stronger. Increased contractility. Your stomach, this is not the time to digest food, so GI motility is slowed down. When you just think about it, once they give you any presentation, you can tell, okay, is this something I'm going to want to do? A person will do when they encounter a lion or they are chased by a lion. If the answer is yes, then that is a sympathetic response. On the flip side, we have the parasympathetic response. The parasympathetic response is what you experience or how a person would act in, let's say you just finished eating, you're so full and you just want to rest. That is parasympathetic. You've eaten, you are so full, what do? You want to rest. That is when most people want to use the bathroom. Because you're so full, you have to open your sphincters so you could pee. Whether your anal sphincters, your urinary sphincters, your GI sphincters, you need to digest food. That's when you have increased GI motility. These are things that will happen when a person is full. You probably lie down to sleep. If you're sleeping, your heart rate goes down. Your heart does not need to work a lot. Contractility goes down. Contractility goes down. Your respiratory rate goes down. You don't need to breathe too much. You're sleeping. Your muscles don't need a lot of oxygen. Respiratory rate goes down. By the way, respiratory rate will go up under sympathetic response. So you see, you can just keep thinking of all of these things and you can figure out a lot of the things that are going to occur in people when they have parasympathetic or sympathetic responses. The next thing you want to think about is, where do we get these systems from? When we say parasympathetic or sympathetic system, what exactly are we talking about? We have a sympathetic outflow. When we look at the spinal cord, let's take this to be our spinal cord, somewhere around T1 to T12. This is what it looks like if we think of the spinal cord itself. Think of your spinal cord, we have an anterior horn, a posterior horn. Around the region of T1 to T12, we have a lateral horn. We have a lateral horn. If you get any image online, you're going to see that. This is the anterior horn. This is the dorsal horn or posterior horn, doesn't matter much. And then we have this lateral horn along the thoracic area, right? Around the thoracic vertebra or thoracic segment of the spinal cord. And then we have those nerves that emerge from the lateral horn. Those nerves are what give rise to sympathetic outflow. Those nerves leave the spinal cord and synapse somewhere here. So they come in here. Imagine this is the nerve. It synapses with another nerve that takes the information to the rest of the body. Okay. This area where we have these nerves synapsing. So imagine we have one nerve comes here, synapses with another nerve. This area is what we call the sympathetic ganglion. Sympathetic ganglion or sympathetic chain. We have it on both sides. Okay, so sometimes they're going to talk about the paravertebral ganglion. Whatever nomenclature they use, it's all the same thing. What do you need to know? Notice that these nerves that leave the spinal cord and synapse here are much shorter than these ones that are going to go to supply maybe an organ somewhere. Notice that these nerves that leave the spinal cord and end up in this ganglion are going to be called preganglionic fibers. Okay, that are the preganglionic fibers. That means fibers before the ganglion. The fibers after the ganglion are post-ganglionic fibers. And they may ask you, you just need to know that preganglionic fibers are short. Generally speaking, post-ganglionic fibers are long. The next thing you want to remember is that within these synapses, the neurotransmitter that these two nerves use to communicate, the neurotransmitter that these two nerves use to communicate is acetylcholine. The neurotransmitter that these two nerves use to communicate is acetylcholine. And the receptor within the ganglion is your nicotinic receptor. The receptor within the paravertebral ganglion is the nicotinic receptor. The neurotransmitter that mediates connection or communication between these nerves is acetylcholine. That is important. That is frequently tested. Remember, I didn't mention this earlier. You want to know that when we talk about parasympathetic, now this is where a little conflict may come in, so follow me closely. When we talk about the parasympathetic nervous system, the neurotransmitter associated with this system is acetylcholine for the most part. When we talk about the sympathetic nervous system, the neurotransmitter associated with this are the catecholamines. Okay, they are your catecholamines. So we are talking about epinephrine and norepinephrine. Take note that epinephrine, norepinephrine is the same as adrenaline, noradrenaline. All right, so this is the sympathetic outflow. Although we are talking about the sympathetic outflow, within the paravertebral ganglion or the sympathetic ganglion or the sympathetic chain all mean the same thing. The neurotransmitter is actually acetylcholine. The receptor is a nicotinic receptor. However, at the end organs over here, the receptors are sympathetic receptors. We have a number of sympathetic receptors you have to know: alpha-1 receptors, alpha-2 receptors, beta-1 receptors, beta-2 receptors, and even beta-3 receptors. Of all these receptors, alpha-1, alpha-2, beta-1, beta-2 are the most important, so we might as well do away with beta-3 receptors. It's important that you remember the location of these receptors and where you find them in different parts of the body. There are a lot of them listed in your first aid, for example. There are others you need to pay attention to. Not everything is so important, but there are things you have to know before you go into your exam. So we just talked about sympathetic outflow. There is one specific sympathetic outflow that is pretty unique and that you have to know. It is the sympathetic outflow to the adrenal gland, the sympathetic outflow to the adrenal gland. The nerve that leaves the spinal cord to the adrenal gland leaves the spinal cord. It does not synapse within the ganglion. It goes straight to the adrenal gland and directly to the adrenal medulla. So it goes directly to the adrenal gland and synapses behind or at the adrenal medulla. Take note of this. Even at the adrenal medulla, the receptor where this nerve communicates with the adrenal medulla is still acetylcholine receptor, nicotinic receptor. However, the adrenal medulla, of course, is going to go ahead to secrete, especially epinephrine and norepinephrine, right? So you want to remember that the nerve, the sympathetic nerve to the adrenal medulla does not synapse within the ganglion. It goes straight to the adrenal medulla, stimulates nicotinic receptors. That's a question on your exam. It is still nicotinic receptors, although we are not dealing with the sympathetic chain now. And then the adrenal gland in turn releases epinephrine and norepinephrine. And then epinephrine would reach out to all of these receptors and you want to know them. Then we'll talk about epinephrine and norepinephrine very briefly before we talk about acetylcholine for 1 second. Epinephrine is secreted from the adrenal medulla. It's almost like a hormone into blood. Norepinephrine, on the other hand, for the most part, is released from nerve endings. So these nerve endings are these organs, stimulating alpha-1, alpha-2 receptors, and beta-1 receptors. You want to know this. We'll come back to these guys. Beta-2 receptors are not innervated. Okay, beta-2 receptors are not innervated. Now, I told you that norepinephrine is produced from nerve endings. Norepinephrine is going to affect alpha-1 receptors because alpha-1 receptors are innervated. It's going to affect alpha-2 receptors because alpha-2 receptors are innervated. And it's going to affect beta-1 receptors because beta-1 receptors are innervated. But no effect on beta-2 receptors because beta-2 receptors are not innervated. Epinephrine, on the other hand, is going to affect alpha-1, alpha-2, beta-1, and beta-2 receptors. Because epinephrine is secreted directly into the blood, it does not need any nerve ending. It can influence all of these receptors. You have to bear that in mind, right? You have to bear that in mind. And that is the simple summary of the sympathetic outflow. Then we go ahead to talk about the parasympathetic outflow. We have 2 sections we need to look at to talk about the parasympathetic outflow. Parasympathetic outflow is from the CNS, cranial nerves. Let's say this is the brain and the brain stem, right? Especially the brain stem, where those nerves emerge from. We have some nerves, cranial nerves, that carry parasympathetic nerves, parasympathetic activity, okay? Cranial nerves carry parasympathetic activity. They do parasympathetic supplies. For example, if you think of cranial nerve 3, oculomotor nerve, you'll notice that your cranial oculomotor nerve carries the parasympathetic neurons from the nucleus of Edinger-Westphal. If you think of your vagus nerve, oh my God, vagus nerve has a lot of parasympathetic connections and communications to a good bunch of the body. All right, so you want to think of cranial nerves as carrying parasympathetic, and then we have S2 to S4 part of the spinal cord also having some parasympathetic components, having some parasympathetic outflow. You want to remember those. We've talked about the receptors that are sympathetic, alpha 1, alpha 2, basically alpha and beta receptors. Now let's talk about receptors that are parasympathetic. Receptors that are parasympathetic. Parasympathetic receptors. Remember, first of all, that the neurotransmitter is acetylcholine. The neurotransmitter is acetylcholine. So what are the receptors that are parasympathetic? They are muscarinic receptors and nicotinic receptors. Muscarinic and nicotinic receptors. Muscarinic receptors are M receptors. You'd have heard of M1, M2, M3, up to M5. So we have M1 to M5 receptors that are muscarinic receptors. What you want to know about these muscarinic receptors is that these receptors are G protein coupled. They are G protein coupled receptors. These muscarinic receptors are G protein coupled receptors. You want to remember that these are the receptors that do all the parasympathetic effects in the body. So increase secretions, for example, right? Increased secretions. It's when people have eaten and they are full that they secrete GI hormones or GI enzymes, for example. This is when people sit down and then they begin to reflect on whatever, begin to cry, exocrine glands, right? All your exocrine glands are stimulated in these cases, right? So you could have exocrine glands stimulated, like your tear glands, your salivary glands. That is when people salivate. That is when people release tears and all of that. We also find some of these muscarinic receptors on the heart. For example, this one may be important. M2 receptors are found on the heart. So muscarinic receptors, the point is that muscarinic receptors are parasympathetic receptors. Then we have nicotinic receptors. They are also parasympathetic receptors. But what I want you to remember is this. We have the nicotinic N receptors and the nicotinic M receptors. The nicotinic N receptors are found where? In the paravertebral ganglion. In the paravertebral ganglion, right? The sympathetic chain, we just talked about that. And the NM receptors are found at neuromuscular, easy to remember. They are found at neuromuscular junctions. They are found at neuromuscular junctions, which means that they are influencing muscular activities, skeletal muscle activity, right? If you stimulate those receptors, the muscles contract or relax and all of those things. So those are what sympathetic activities are, what parasympathetic activities are. The other thing that is very important that you remember is the location of these receptors. Where do we find these M1, M2, and so on? Where do we find, we already know where we find the NM. This is actually important for your exam. We'll talk about diseases where you have damage to those receptors, anti-acetylcholine receptor antibodies, for example. I hope you're thinking of myasthenia gravis already. All right. Then we want to know where you find alpha 1 receptors. You want to know where you find alpha 2 receptors. You want to know where you find beta 1 receptors. You want to know where you find beta 2 receptors and what their function is. There's a lot of activity that all these receptors do that you may need to know, but there are some of them that are very essential. I'm going to mention those ones. You can read your first aid, for example, you're going to see a lot of activities that these receptors do. You need to know what alpha 1 receptor does. Alpha 1 receptors are found on vascular smooth muscles, blood vessels, basically. They're found on blood vessels. And what do? Alpha, sympathetic vasoconstriction. They're found on blood vessels, causing what? Vasoconstriction, because they cause those smooth muscles to contract. They're also found in the eye. It is the alpha-1 receptors that when they are stimulated, there is pupillary dilation. So you can imagine that we find them on the pupillary dilator muscle of the eye, right? We find them on the pupillary dilator muscle of the eye. And what are they doing here? They're causing mydriasis. You have to remember that. And then we find them in sphincters. What are they doing in sphincters? They're causing urinary retention, for example. In the urinary sphincter, they're causing constipation in the GI. So you have to think of them like that. If you think of them like this, you know where it is and what the effect is. Where is the receptor? What's the effect? We talk about alpha-2 receptor. You want to know that alpha-2 receptor is an autoreceptor. That was a question on one of the MDMs. Alpha-2 receptors are autoreceptors. They are autoregulatory. We would see them when we draw the, what do we call it? The nerve endings. We would see alpha-2 receptors. We can find alpha-2 receptors. You want to notice that they are the receptors that are located on both presynaptic and synaptic endings. They are located on both the presynaptic and the presynaptic nerves, and we are going to see this eventually. They have many other effects that we don't really care about at this point. Beta1 receptors are found on the heart, and what they're doing here is basically increase heart rate, increase contractility, basically inotropy, dromotropy, things like that, right? These guys also increase blood pressure ultimately, so they can increase renin release. Increase renin because we find them on those tissues of your kidney involved in renin production. All right, and then we talk about the beta2 receptors. There are two main places you want to know that they're also found on blood vessels. And finding them on blood vessels, they're actually causing vasodilation, unlike the alpha-1 receptors that are causing vasoconstriction. The other thing you want to remember is that we find them in the smooth muscles of the bronchi. So bronchial smooth muscles. And here they're going to be causing bronchodilation. Here they're going to be causing bronchodilation. So these are the two most important areas. Of course, we have other things that they do. Maybe a third one would be uterine smooth muscle. And of course, uterine, nobody, for now, we are not going into that. These are the most important things, more frequently talked about or tested or whatever that you need to know. So they cause vasodilation, they cause bronchodilation. They have effect on insulin. Maybe they, let's say they increase insulin release. If they're increasing insulin release, they're increasing potassium uptake into the cells. All of those things are there, but for now, you must know these ones to at least know most of the activity of a lot of the drugs you're going to be looking at in your general pharmacology section, right? So those are beta1, beta2 receptors. We have beta3. Beta3 does relaxation of the bladder. I think that's the most important one. It does bladder relaxation. And then, of course, it does lipolysis, thermogenesis. I don't care about those ones for now. But you should know these ones, especially. If you don't know anything, you must remember this part about these receptors. And then we talk about the muscarinic receptors. We have M1, M2, M3, M4, M5. For the most part, I'm going to be talking about the M1, M2, M3s. M1 is CNS. It's found in the CNS. M2, we already said, is found especially on the heart. And then your M3 receptors does a lot of secretory things, okay? So your salivary glands, your respiratory airway, all of those things. Not a very important thing, forget about it. So M1, M2, M3 receptors. And then, of course, we have other receptors that we could be talking about, histamine receptors, dopamine receptors. If you look at the first aid, you see that they have a lot of those receptors lined up there. This is what you need to remember. This is what you need to remember. Now, the other thing you want to know is that, of course, we already said that neurotransmitters act on these receptors. When these neurotransmitters act on these receptors, what is the outcome? That's what we have to ask. When neurotransmitters act on these receptors, what is the outcome? When we have a cell like this one, we have this receptor. Let's say this is alpha-1 receptor. When you stimulate alpha-1 receptor, okay, then what? There is a second system, okay? What we call second messenger systems. We have second messenger systems, second messenger pathways, doesn't matter. This alpha-1 receptor activates another set and series of activities within the cell that result in contraction, relaxation, whatever it is that that particular receptor is supposed to do. And this is another important part of general pharmacology that you have to know to the best way possible. You have to know this. These second messengers are mostly G proteins. We'll see a lot of second messengers later on. You have second messengers in a lot of places, but these second messengers are G proteins, and we have different components of G proteins. G proteins can be GQ, they can be GS, they can be GI. GQ, GS, GI. I like to think of S as stimulatory. I like to think of I as inhibitory. I like to look at Q as so-so. Don't stress about it. Just know that GQ, GS, and GI. Now, you want to know what series of events, and you want to know them exactly in that manner, because when they ask this question, they can ask you about any part of the G protein pathway, and they would need you to actually remember them. We'll start with the GS pathway. When the GS receptor is stimulated, it activates adenylate cyclase, an enzyme. Adenylate cyclase is an enzyme. And what this enzyme does really is catalyze the conversion of ATP, right, to CAMP. It catalyzes the conversion of ATP to cyclic AMP. Now, this is what I need you to do. One way is I need you to memorize the pathway exactly as it is on display here, or exactly as it is in your first aid. Actually, they're just about the same thing. You will need to know exactly the pathway, or you need to know where each thing is. So you need to know that GS is on the same place as adenylate cyclase, as the same place as CAMP. Okay, you need to have that, sort of know that in your mind somehow. So these GS receptors, when activated, are going to activate or turn on adenylate cyclase activity, and adenylate cyclase is going to convert ATP to CAMP. CAMP is going to go on to activate protein kinase A. It's going to go on to activate protein kinase A, and protein kinase A, in turn, will do two things. How do I put that? Protein kinase A is going to do two things. One is increase calcium influx, okay? Increase calcium influx, especially on the heart. The second thing that protein kinase A does is inhibition of myosin light chain kinase. It's going to inhibit myosin light chain kinase. Myosin light chain kinase is found on smooth muscles. That's the enzyme that actually causes vasoconstriction. So by inhibiting myosin light chain kinase, protein kinase A is actually going to be causing vasodilation. Okay, so you want to remember that. And then we have to ask what receptors are GS coupled, but we'll come to that in a bit. The second receptor we have is GI receptors. All I want you to remember is GI is an inhibitor of the GS system. GI is an inhibitor of the GS pathway. So it inhibits adenylate cyclase activity, therefore it will decrease CAMP. If it decreases CAMP, it's going to decrease the activity of protein kinase A, so it's going to decrease calcium influx into the heart. It's going to block the inhibition of myosin light chain kinase, so there'll be more activity of myosin light chain kinase. So GI is just the reverse explanation for what GS is. So let's look at what receptors, or what receptors are GS coupled. So alpha-1 receptors, all these receptors we are talking about all along are coupled. I showed you somewhere here. So we have this receptor coupled to this guy inside the cell. So when we say receptor coupling, this is what we are talking about. So what receptors, this is the G protein inside here. So what is coupled to GS. Who is coupled to GI? You need to know that specifically. M2 receptors, alpha-2 receptors, and D2 receptors, dopaminergic receptors, are GI coupled. M2, alpha-2, D2 receptors are GI coupled. Like I call them the MAD2s. The MAD2s, they are inhibitors, right? They are inhibitors, MAD2s, M2, alpha-2, D2. You want to remember that. So who is GS coupled? This is a little more difficult to remember, but your beta-1 receptors, your beta-2 receptors, your H2 receptors, your V2 receptors, and I think your D1 receptors are GS coupled. You don't have to memorize that. It's easier to remember what is GI, and you will find out what is GQ. Once you remember what is GI, what is GQ, you just know everybody else at GS coupled. That makes the work easy. Finally, we have GQ receptors. GQ receptor. GQ receptor activates the enzyme phospholipase C. It activates phospholipase C, and phospholipase C in turn converts PIP2 to two enzymes. It converts PIP2 to DAG, diacylglycerol, and inositol triphosphate, IP3. You want to know that. DAG, diacylglycerol, goes on to activate protein kinase C. Take note that protein kinase C is on the second pathway. Protein kinase A was for GSGI. Protein kinase C is associated with GQ receptors, okay? Pretty easy, makes sense, I think. And then IP3 is the guy that increases calcium in this case. So IP3 directly increases calcium influx, and this is going to result in contractility, increased contractility. You want to know that calcium can, of course, also increase protein kinase C, but that is not very important. So what receptors are GQ coupled? What receptors are GQ coupled? H1, alpha 1, V1, M1, I think M3. Yeah, those are the receptors that are GQ coupled. H1, A1, V1, M1, M3. There's a mnemonic in your first aid. I don't know how it goes. I just know it's like half MM or something like that. Half N, M, and M, something like that. H1, alpha 1, V1, M1, M3. I just know how it is. You can use the mnemonics if you are a mnemonics somebody, okay? And these are the receptors that are GQ coupled. You want to remember them. Very important when they ask you these basic questions that you can actually recall them. And there you have it. Those are basic sympathetic and parasympathetic activities. They are receptors where you find them, and they are receptor coupling. Maybe we are going to talk about the drugs. I'm not sure we'll go into all of that. If you understand this pathway, I think you can make sense of a lot of the other things that follow the sympathetic or autonomic drugs and all of that you're going to see in your first aid. If you cannot do that still, put it in the comments, let me know. Okay, so what now? And then I'm going to go ahead and make more of the videos, maybe teach the whole of general farm, who knows? I'm probably wanting to do that. And I'll put it out for you guys. So this is where we're going to stop. Remember, this is autonomic nervous system. Sympathetic activities are what happens when a lion chases you. Parasympathetic activities are what happen when you're well fed and you just want to sleep and you have enough time to use the bathroom. Think of it in that way and you will not forget it. Remember where these receptors are found. Remember the function of each of these receptors. Alpha 1 is causing vasodilation. Beta 2 is causing bronchodilation. It's also causing, alpha 1 is causing vasoconstriction. Beta 2 is causing vasodilation and bronchodilation. And then when you've mastered that, try looking at medications and all of that. I think I'm going to make a video where I actually take these medications, or I go through questions on general farm related to this, and we're going to break down each of them. I think that should be the next video. What do you think? Let me know in the comments, and I'll see you guys soon. Okay.